Today we are going to look into a research microscope or binocular microscope, which is generally used in a laboratory for research purposes or in a diagnostic lab for diagnosis of different microorganisms. So starting off with the parts in the microscope, at the very first we are in the top we have the objective lens, which is used to visualize our sample or specimen in the slide. And the objective lens is connected to the nose region with the neck region. The nose region have different lenses of different magnifications, like from 4 into 10 into 40 into and then 100 into magnifications. The neck region is also attached to the arm region. The arm region has two knobs present in here. The bigger one is called a course adjustment knob, which controls the upward and downward movement of the platform present here. And then there is the smaller knob, which is called the fine adjustment knob, which controls the focus of the image we are going to see. So in the platform here we have this tiny mechanism present, which is used to hold the slide in its place, and this tiny mechanism can be controlled by the knob present in the right side. The upward knob is one which controls the forward and backward movement of the mechanism, and the lower smaller knob controls the lateral movement of the mechanism. So why are we controlling the mechanism? So we are controlling the mechanism to bring the slide into the place, and the places in the platform there is a small hole present. The hole is used as a source of light. The main source of light is present down below here, and this source of light can also be controlled by a knob present here. So once we have placed our slide in the platform in its place, we will use the coarse and fine adjustment knobs to properly bring the sample specimen into a focus. And once the specimen is into focus, we will use the objective lens to observe our slide.